Biostimulation, a tool for reproductive management in small ruminants. We'll be discussing the different points like what is biostimulation, what are the different pheromones, its types, sources, what are the sensory pathways of pheromones, what are the roles of biostimulation in sheep and goat reproduction, conclusion, future line of work, and then we may discuss a few other things after the presentation. All of us know that India is the country which is having largest livestock population of 536 million numbers. And in the last livestock census, it has increased by 4.8%. So as far as sheep and goats are concerned, these are the earliest discovery of the mankind for harvesting of meats. And however, this human population took some more time to understand the value of milk, hair, and skins. And this sheep and goat is having very good important role in socio-economic development of the marginal and poor farmers throughout the world. So this sheep and goat are easily adaptable, and it can be kept in intensive production system, that is zerograzing system, and it can convert their feed into highly nutritious milk and meat very efficiently. If you see the worldwide position, so this milk type goats are mostly found in the temperate zones of the world, and this dual type and meat type are found in this subtropical and tropical countries of the world. So goat is having no any social or religious taboo as far as this Indian religious and cultural concern is there. So it is accepted by all the people in India and throughout the world also. And therefore, this goat meat has been considered as national meat animal in India, as there is no social taboo with regard to this. Contribution of this small ruminant in total livestock population is about 40% in India. And as we know that this total sheep population is 74 million, and the state which is having highest sheep population is Telangana. And similarly, the goat population is 148 million, and Rajasthan is having the top goat population in India. As far as this milk production is concerned, out of total 230 million tons of milk India is produced. So the goat is having little contribution in milk production, that is around 3.5% in India's total milk production. But in meat production, as there are 9.7 million tons of meat is produced in India last year, and this sheep and goat contributed around 25% in the total meat production in India. So the wool production is somewhat, we can say, in declining trend, as there is a low demand of this wool, and there is availability of synthetic wools are also available. So these are the different ram, ewe, and lamb which produces wool production. So now we are coming to the actual topic, that the livestock productivity in the tropical region of the world, that is India, is low relative as compared to temperate climatic conditions. And there are many reasons for this, which is mainly due to this environmental stresses in the tropical countries like India, poor nutritional status of the animals, and other managemental problems which are there in the farm level. So these various attempts have been tried to control this animal reproduction in small ruminants by use of different types of hormones and its analogs. But although these hormonal treatments are highly effective for control of the animal reproduction in small ruminants, but due to growing concern of the clean, green, and ethical environment, and the uses of drugs and exogenous hormones to be reduced. So besides this, hormone and analogs are also not easily available in the rural areas and too much expensive for the Indian farmers. This harnessing of the reproductive potential of a small ruminant, it may require some development, some management strategies, which is biostimulation, as this biostimulation can effectively improve the reproduction in small ruminants. So this biostimulation technique is a potential useful and practical way to improve the reproductive efficiency in sheep and goat in the tropical countries like India, as It can hasten the puberty and sexual maturity in the small ruminants. It can terminate this seasonal anestrus and shorten the postpartum anestrus in the small ruminants. And it also enhances this sexual performance of a ram or buck, that is male animal also. It is not like that only the male animals can affect the female animals. Females can also affect the performance of male animals. We'll discuss this later. So before I proceed to this topic, let us have a look on this estrous period of the estrous cycle of the goat. That is between one estrous period to the next estrous period. It is called estrous cycle, and there are two phases, that is luteal phase and follicular phase. So this proestrus and estrus are considered as follicular phase of the estrous cycle, and metaestrus and diestrus are luteal phase. So proestrus is the period where there is significant rise of the estrogen level in the blood, and which is produced by the developing follicles. And as the estradiol or estrogen level reaches a certain level, the female used to exhibit the stress. They show the stress symptoms, and then there is ovulation. And after the ovulation take place, the follicles are transferred into luteal cells, and there is formation of corpus luteum. And in the diestrus phase, this corpus luteum is fully functional, and it secretes the progesterone hormone from there. So this the luteal phase also may be considered as into three phases, that is early luteal phase, mid luteal phase, late luteal phase, that we'll discuss. So these are the reproductive parameters of sheep and goat, which is normal reproductive parameters like puberty, estrous cycle, estrous length, time of ovulation, gestation period, all these things. Now coming to the topic, that is biostimulation. So biostimulation is a pheromonal communication that plays an important role in sheep and goat behavior and reproductive process. Means it's a communication between the male and female with mainly pheromone, but there are some other cues also. So this stimulatory effect of male on a female cyclic activity through genital stimulation, pheromones, or other less defined external cues is known as biostimulation. So this biostimulation concept was given by Heap in 1901. It is the early 19th century. And from that time, he told that the presence of male could hasten the onset of puberty, sexual maturity, ovulation, induction, and reduction of postpartum anestrus in females. So from that time, this the biostimulation concept came, and from there the research work started in these fields, and slowly the research was going on. And during 1980s, the maximum research on this biostimulation, buck effect, and pheromones took place, and what we are progressing nowadays also. So this pheromone word was given by this Carlson and Lusser in 1959. So where it affects actually this biostimulation? So it can affect male, can affect female, female can affect male, or female can affect female also, and sometimes male can affect male also. So the presence, this male also affect the male. So how it works? So the different extoreceptive cues, they plays a very important role in male-female interaction, and that includes, so this is different extoreceptive cues like olfactory, visual, auditory, and tactile. These are the four extoreceptive cues which plays important role in interaction with male and female. And this olfactory is the most important one, because it involves these pheromones. So it is also said that a high percentage of females respond to male stimulation when all cues are active. So these are some of the examples to understand better that this olfactory cues, this is very famous in this musk deers, where musk glands are there. They used to secrete some pheromones, and there is communication between the male and female with the help of this musk glands. So visual communication is very important in case of peacocks, auditory is in case of this elephants, and tactile is in case of horses. So all these extroreceptive cues, that is olfactory, tactile, auditory, and visual. So these are what they are doing. They are actually giving some signals to the brain, and from there is communication to the hypothalamus, and there is release of gonadotropin releasing hormone, and that is responsible for the release of FSH and LH, follicle stimulating hormone and luteinizing hormone, and they are actually causing this growth and development of the follicles and the ovulation of the follicles, all these things. So in short, how it works. Different many advantages of biostimulation is also there. It is clean, green, and ethical animal production, accurately control these reproductive events, and it is cost effective also. So to study about the biostimulation, it is very important to know the pheromones and its types, because out of these 4 extroreceptive cues, mainly the pheromones are the major one. So thus understanding the role of pheromones could potentially important economic in addressing some of the problems associated with the small ruminant production in the tropics. So coming to the pheromones, its types and sources. So what are the pheromones? Pheromones are actually these airborne chemical substances which are secreted externally in the urine, feces, or cutaneous, or by the cutaneous glands. It causes a specific reaction in a receiving individual of the same species. It is said same species, but 1 or 2 studies I'll show here that it affects on some other species also. Suppose goat can affect sheep also. So it initiates a specific behavioral or physiological changes in the recipient's endocrine and reproductive system of the same species, or sometimes other species also. It is classified on the basis on 2 types of pheromones, that is signaling pheromones and priming pheromones. So these are the 2 types of pheromones. That is, first is signaling pheromone. Signaling pheromone is actually it causes some behavioral changes in the animals. So it involves this olfactory cues. It transmits specific information. And we have seen this Flehmen reaction in case of male animals when they detect some females in estrus due to pheromones in the urine or vaginal secretions. So it shows a Flehmen reaction. It is one example of signaling pheromone. It is proved to be helpful in detection of estrus in females. And priming pheromone, what it does, it causes some physiological events in the endocrine or reproductive system. Means it causes hormonal changes in the animal. Signaling causes behavioral changes, and priming causes this hormonal changes in the animal. It also involves this olfactory cues, and it causes some physiological responses in the animal. So priming pheromones from these male influence the induction of puberty, termination of seasonal anestrus, shortening of postpartum anestrus, and affects ovarian activity. So these are the sources of different pheromones. That is, in case of goat, it present in urine, mohair. In case of sheep, it is available in wool, waxes, or urines. Now, this sensory pathway of pheromones. So this sensory pathway of pheromone, this pheromone can exert profound effect on the this reproductive activity via the hypothalamic system that generates the pulses of gonadotropin releasing hormone. Actually, these pheromones which are received by the nasal cavity of the mammals, and there are 2 types of organs are there. That is main olfactory epithelium and vomeronasal organ. So this main olfactory epithelium, these are the these are useful in generalized type of functions. Means they detect some general type of molecular analyzer for various odors, environmental chemicals, having no predetermined meaning. Means general, it receives this general type of information. But vomeronasal organs receives specialized type of informations like pheromones, specific pheromones. It carries some information about the gender, reproductive, or dominance status, or specific informations are received by these vomeronasal organs. So here we see in a through one diagram that pheromones, how it works, stimulatory pathway. So you see this is the picture of goat, and this male here we see the pheromones are perceived by the vomeronasal organ. Then this contains some specific receptors, neuron receptors, which causes some stimulate this cascade of neuroendocrine responses in the female. And here we see this there are two parts, that is central nervous system and reproductive system. So in central nervous system, we see this male stimuli is there, pheromones are there, which are received by the olfactory senses. Then it goes to hypothalamus, it releases GnRH. Then from the anterior part of the pituitary, it releases this FSH-LH, and it acts on the ovary of the animal. So this is how this pheromones work. This is one example of primer pheromone. So these primer pheromones are released by the males, and it is received by the female goat and through the olfactory receptors. Then it causes GnRH pulse generator, and there is reproductive endocrine system. There are some changes are there. Now we are coming to this role of biostimulation in sheep and goat reproduction. So mainly this sheep and goat reproduction, there are mainly four types of roles are there. That is effect on puberty and sexual maturity of the animals, then effect on intensity of estrus and its synchronization, then reduces postpartum anestrus and enhances male sexual performances. So first we are discussing that effects on puberty and sexual maturity of the animals. So this very early in 1950s, this scientists have told that in small ruminants, this ram effect or buck effect was reported to hasten the onset of estrus activity in females. And this priming pheromones from the male influences the induction of puberty in female sheep and goat. This was also observed during 1980s. And this buck exposure potentiates the effect of PMSG inducing estrus in prepubertal dose. So this in this research, you can see that the estrus response and interval after treatment to estrus of prepubertal goats treated with some hormonal treatment and stimulus. Here we can see that there are three groups. There were three groups. One is hormonal treatment group, where they have used synchromet B and PMSG hormone, and another group, along with hormone, they had used buck effect, and the third group only buck exposure was there. So we can see here clearly that in prepubertal dose, so when this buck effect was added with the hormonal treatment, there was more number of animals in estrus. 91% of the animals were in estrus by this treatment, as compared to 66% in only hormonal treatment. And in case of only buck exposure, there was very less number of animals were in estrus. However, this interval to onset to estrus was at par in this both the treatments, and more in case of buck exposure. Now the effects on intensity of estrus and its synchronization. So during the normal breeding season, the introduction of males enhances the onset of estrus cycle in seasonally anestrus use and dose. Normally, it is mainly there that this male effect or biostimulation works for this animals which are in anestrus condition for a long period of time due to seasonal variables. But nowadays, it is also the works are also there. The biostimulation effect on this cyclic animals also. Now, the presence of large number of ewes in the onset of estrus can also stimulate this ovarian activity in seasonal anestrus ewes. So this is called female to female effect. Means when a large number of ewes are in estrus, some animals which are not in estrus condition, they also come into estrus due to the effect of female effect, and which are mediated by this olfactory, visual, and auditory cues. Now, this male effect was as effective as synchronization treatment involving some hormone, synchromet B. So here we can see this in this research. This 4 treatments were there. So one treatment is only buck teasing. Another is this hormonal treatment that is called Synchromate B, and buck effect 1 day after treatment. Means there was no combination of this hormonal and buck effect. After completion of hormonal treatment, after 1 day, they have introduced buck. And in these 2 days before end of treatment, hormonal treatment, and this 1 day before end of treatment. So these two groups where this buck effect was clubbed with the hormonal treatment, means before end of hormonal treatment, bucks were introduced, they gave better result as compared to the group where the buck effect was introduced after end of hormonal treatment. And only buck teasing was also as effective as the combination of hormonal treatment and buck effect. Means these three groups worked at par as compared to this group where there was no combination of buck effect and hormonal treatment. So we can say that buck effect is equally effective as that of hormonal treatment and buck effect in combination. So this is the study which was conducted by me during my master's degree program. So here also we have worked on these cyclic animals for synchronization of estrus in cyclic dose. So here what we did that there was three treatment groups. One was control group where there was no any treatment, neither hormonal treatment nor this buck effect. In second group, we had used hormonal treatment, that is PGF2 alpha, 2 injections at 11 days apart. And after second injection, means first injection, suppose we had given on 1st of 1 month, and on 11th, when we gave this second injection of PGF2 alpha, on the same day, we started the third group, that is buck effect group, and from second injection day, we have noted the this observation. So onset of estrus was at par in hormonal treatment and buck effect, although this range was more in case of buck effect. So in PGF2 alpha group, although the average is 64 hours onset of estrus, but the duration was 50 hours to 74 hours. But in case of buck effect, the duration was 20 hours to 120 hours. Means the range was bigger. It took 5 days time, but it synchronized as compared to hormonal treatment where 3 days was there. So duration of estrus was at par, and you can see this degree of synchrony. It was obviously in case of hormonal treatment, it was better. Out of 6, 5 came into estrus in within 72 hours, and 1 remaining animal after 72 hours. And in case of buck effect, 50% animal came in within 72 hours and 50% animal after 72 hours, means but within 120 hours. So it took 5 days in buck effect, and it took 3 days in case of hormonal treatment, but it synchronized. So this study also correlated with the hormonal level of the animals. Here we can see this hormone of estradiol 17 beta and progesterone level of the animals. So here we can see that this hormonal treatment group, this estrogen level increased and progesterone level decreased as after 0 day was the day of treatment. After day of treatment, after 1 day, it crossed each other. And in case of buck effect group, we see it also crossed, but it took 1 day more. Means on second day, it crossed each other. And in case of control group, you see it was at par. We had also studied this behavioral responses of animals during the buck effect and hormonal treatment. Although this bleating and tail fanning was observed in maximum animals, but there was no significant difference in the behavior of animals in buck effect or hormonal treatment. So this is the effect on postpartum anestrus. So in postpartum anestrus, in domestic animals, priming pheromones from the male influences the termination of seasonal anestrus and shortening of postpartum anestrus. Along with this, it is also helpful in increasing intensity and duration of the estrus in animals. So here you can see these 2 groups were there, RAM exposure and no RAM exposure. So in RAM exposure group, you see the number of females ovulating were 100%, and no ram exposure group, there was 50% animal ovulated. So the ram exposure is helpful in animals to bring into cyclicity after parturition. Now, effects on male sexual performances. So pheromones are also released by the females during the estrus, thus stimulating the sexual behavior and endocrine function of the males. And this sexual stimulation technique that improves male sexual performances have been reported in several farm animals also. And here we can see that this sudden introduction of female of estrus ewes into the group of rams, when there is sudden introduction of ewes which are in estrus into the group of rams, there is changes in the behavior and endocrine state of the male. That is called female effect. So this sexual interaction of male with female tend to increase the plasma level of the testosterone, and which is further associated with the rapid rise in luteinizing hormone concentration in the animal, which is responsible for the libido in males. Yearling rams kept close to the ewes in estrus for few months, that is 6 months, were found to have larger testes, high plasma testosterone level, and greater sexual activity. When these rams, which are about 1 year of age, when they are kept in use in estrus, these changes were seen. So exposure of male to female in estrus, viewing this when these males are exposed to females which are in estrus, when they see the females copulating, means when there is mating, they are seeing, and this periodical changes of female has been reported to enhance the male sexual performance in several species. So these females are also affecting the behavioral changes in the males. Now, some of the factors that affect the male effect that we'll discuss here. So effectiveness of ram and buck for stimulating the ewe to ovulate means when you see this isolated ewes were there when teased with ram and when teased with buck. So ewes means one species, it is ewes sheep. When the female of sheep are teased with ram and buck, so it is obviously the by value we can see more number of animals ovulated by rams, but buck also ovulated 57%, which is at par. Significantly, there was no difference. And when it was isolated, only 7% was there. Means buck and ram both are equally effective in stimulating ewes to ovulate. Now, this effect of social dominance of female goats on response to male means the dominancy level of the animals. In case of sheep and goat, there is some hierarchy they develop. Some are high rank, medium rank, low rank. So this high rank or medium rank animals are able to remain in the more close vicinity with the males, and there is it has been observed in the ovulation. Means the ovulation time was less in high rank or medium rank female as compared to low rank females. Similarly, in case of conception rate also, this high rank female was able to conceive early as compared to medium rank or low rank animals. Here also we can see this subordinate animals and dominant animals. So you see this number of goats ovulating were significantly higher in dominating group as compared to subordinate group. So dominancy and subordinate also have effect on this result. So in earlier days, when this biostimulation study was started during the early 90s, they assumed that this complete segregation of the animals with regard to odor, sight, sound, and touch is necessary for at least 3 weeks duration, and 500 to 1000 meter distance from male was thought that this is required for to have the male effect. But the latter researches showed that this previous isolation of female from male goat may not be necessary to stimulate their reproductive activity by the male effect if sexually active males are used. So here some of the research are there. So effect of previous isolation of female Mexican Creole goats from male effect by sexually active bucks. So here you can see this one group was there where the females were in contact with the vasectomized buck before male effect. And in this group, isolation group, females were completely isolated from the buck. But when the exposure period started in both the group for 15 days, so you see here 9 out of 11, 10 out of 11 came into estrus and ovulated, and the interval from introduction of buck to first estrus was 2.3 days and 1.9 days. So there was no significant difference in the reproductive behavior of animals when the buck effect was given in the two groups, that is contact group or isolation group. So that it shows that there is no requirement of previous isolation of the animals to have a male effect. Here also you can see two groups were there, isolation group, no isolation group. So the females were isolated from male for 46 days in case of isolation group, and in no isolation group, these females were in contact with the vasectomized male for 46 days before the male effect. So here also we have seen that this ovarian activity, estrus and ovulating percentages, and interval from introduction to estrus days were at par in both the groups. Means whether the animals were kept isolated before the male effect or it were in contact with the male, there is no effect on the result of this ovarian activity or estrus or ovulating of the animals. So this is another thing, that the effect of continuous or discontinuous exposure to male on estrus behavior in Mexican goat. So here two groups were there. In one group, continuous exposure was there, that is 24 hours per day, and exposure period was 18 days. And in another group, every day 16-hour exposure was there. So in this, it is observed that there was no significant difference and the result was at par. Means the days after buck introduction, the percentage of goats in estrus were similar in both the groups in continuous treatment, continuous exposure or discontinuous exposure. But contrary to this, here in this research, what we have seen that when this continuous exposure was given, this estrus onset was significantly early as compared to intermittent exposure, that is 45 hours as compared to 32 hours. This interval from sponge removal to ovulation is also less in continuous group. But the reason for this might be that here in earlier studies, we have seen 24 hours and 16 hours. But here 24 hours and 1 hour. Due to very less exposure time per day, that is only 1 hour per day, this result might have taken that this continuous and intermittent, there is so difference in the result. So this is one another thing, that when adult rams were used and yearling rams were used for this male effect, what is the result? So here we can see this adult ram performed better as compared to yearling ram with regard to marked use, means the percentage of animals in estrus, then conception rate, pregnancy rate, all were significantly better in adult ram group as compared to yearling ram. However, this interval to estrus and use with ovulation were at par. So in this study, this with regard to LH concentration and LH pulses, so here we can see that early luteal phase, mid luteal phase, and late luteal phase. So the after introduction of male, there was significant increase in the LH concentration or LH pulses in the females in early luteal phase and late luteal phase. But in case of mid luteal phase, due to high level of progesterone hormone, the buck effect was blocked in this mid luteal phase. So it is considered, it may be concluded that this buck effect is working in a better way in early luteal phase and late luteal phase, and it fails during mid luteal phase due to high progesterone level. So biostimulation or pheromonal communication plays an important role in behavioral and reproductive processes of sheep and goat. Pheromones in the urine, feces, or from cutaneous glands can be perceived through the olfactory system to elicit both behavioral and endocrine responses in conspecifics, that is same specific. Biostimulation can exert profound effects in the reproductive activity via hypothalamic system and generates pulse of gonadotropin releasing hormone. Biostimulation may be used in females for hastening puberty, sexual maturity, termination of seasonal anestrus, and shortening of postpartum anestrus in small ruminants. The economic benefits of using biostimulation through pheromones to enhance early onset of puberty and the significant reduction of the postpartum anestrus in small ruminants may serve as a reproductive management tool in India, where the livestock production has some constraints. Now, the future line of works may be, these are some of the future line of works, that is isolation, identification, and commercialization of pheromones from males for use in the females is to be done. Urinary cues from the small ruminants are not yet fully explored, which needs attention. A role of non-olfactory cues, that is visual, auditory, and tactile, need to be understood properly. And economic benefits of biostimulation through pheromones need to be calculated.